In a basement lab in Hangzhou, a researcher pulls two complete sets of chromosomes apart from a single goldfish and realizes the orange fish in the tank is not one animal, but two, fused so long ago that no human was alive to see it happen. That single act, separating one genome into its two hidden halves, quietly demolished the story we had told ourselves about goldfish for 1,000 years. We have kept this animal beside us for longer than we have kept almost any other creature, purely for the way it looks. We have bred it into shapes that could not last a single day in a river, and for most of that time we told ourselves a simple story about where it came from. We were wrong about almost all of it. The goldfish is not a tamed exotic. It is not an ancient wild species we plucked from a stream. It is something stranger, a creature assembled from two different fish, then reshaped over a millennium into forms its ancestors would not recognize as kin. The history of the goldfish is written in its genome, in sweeps of selected genes, in a hybridization event so old that researchers had to pull two separate sets of chromosomes apart to read it. This is what the DNA actually shows. For a long time, the goldfish seemed like one of the easier domestication stories to tell. The textbook version went roughly like this. Somewhere in ancient China, people noticed that a common drab pondfish, the crucian carp, occasionally produced an individual with strange coloring. Instead of the usual olive gray, a fish would appear flushed with red or gold. People found these bright mutants beautiful, scooped them out, kept them, and bred them. Over centuries, the story went, that single act of selection, saving the pretty ones, slowly produced the goldfish. One wild fish, one mutation for color, one long, unbroken line of breeding leading straight to the bowl on the windowsill. It was a tidy account, and it had real evidence behind it. The wild ancestor was never in much doubt. Across the rivers and ponds of East Asia lives a small, hardy fish in the genus Carassius, the crucian carp and its close relatives. These fish are unremarkable to look at, dull-colored, broad-bodied, able to survive in low-oxygen water that would kill almost anything else. The crucian carp is famous among biologists for surviving winters sealed under ice in oxygen-starved ponds, switching its metabolism to produce ethanol rather than lactic acid, and breathing it out through its gills, a chemical trick almost no other vertebrate can manage. That is the lineage standing behind the delicate fish in the bowl, one of the toughest freshwater survivors on the planet. Goldfish, when you strip away the fancy fins and the bright scales, have the same basic body plan. The match was obvious, and the historical record reinforced it. Chinese sources describe colored fish being kept and admired as far back as the Jin dynasty, between the 3rd and 5th centuries, when crucian carp with red scales were first noticed and set aside. By the Tang dynasty, between the 7th and 10th centuries, gold, orange, red, and yellow fish were being kept as living ornaments in private ponds, often in the carved stone basins of Buddhist temples, where the practice of releasing and sparing living creatures gave the bright fish a protected place to multiply. By the end of the Song dynasty, the orange ones had acquired the name we still use, and the yellow variety had become so prized that only royalty was permitted to keep it, because gold was the imperial color. Commoners who bred a yellow fish were expected to surrender it, so the broad strokes seemed settled. A wild crucian carp, a color mutation, 1,000 years of Chinese breeders selecting for beauty. The fish was understood as a single species slowly transformed by human taste. The uncertainty was only about details, which exact wild population, which dynasty saw which trait emerge, how fast it all happened. The fundamental shape of the story, one fish becoming another through patient selection, was treated as obvious. The problem was that nobody had ever read the goldfish genome closely enough to test it, and when they finally did, the simple picture broke apart in two places at once. The first crack came not from the goldfish's color at all, but from something buried far deeper in the very structure of its chromosomes. In 2020, a team of researchers affiliated with multiple institutions in China published a study in the Proceedings of the National Academy of Sciences that did for the goldfish what landmark ancient DNA work had done for dogs and horses. Led by researchers including Duo Chen, the team assembled a high-quality goldfish genome, anchoring more than 95% of the genetic sequence into 50 pseudochromosomes, and then resequenced 185 representative goldfish varieties alongside 16 wild crucian carp. It was, by a wide margin, the most comprehensive look at goldfish ancestry ever attempted. They were not just confirming that goldfish came from crucian carp, they were reading, gene by gene, exactly what 1,000 years of human selection had done to the animal. The headline finding was hidden in the architecture of the genome itself. The goldfish carries roughly 100 chromosomes, about twice as many as you would expect. When the team examined them, they found the genome was not one coherent set of instructions, but two. The goldfish, they discovered, carries two distinct subgenomes that had to be carefully disentangled, the legacy of an ancient hybridization event in its deep past. Long before any Chinese breeder ever scooped a red fish from a pond, two different lineages of fish had merged, doubling the genome and fusing into a single animal. This is the same trick, whole genome duplication, that lies behind the bread wheat in our fields and the salmon on our plates. But in the goldfish, it produced a curious redundancy. Two copies of nearly every gene, one free to keep doing its old job while the other drifted, mutated, and occasionally found something new to do. That spare genetic machinery is part of why the goldfish proved so endlessly moldable in human hands. The goldfish was a hybrid creature at its very foundation, built from a genetic collision that predated human involvement entirely. But here is where the story becomes strange. That ancient doubling was only the beginning of the goldfish's mixed parentage. Because when other researchers looked not at the chromosome count, but at the difference between the goldfish's mitochondrial DNA, inherited only down the maternal line, and its nuclear DNA inherited from both parents, they found a contradiction that should not have been there. A separate body of work, examining the polyphyletic origin of ornamental goldfish, compared mitochondrial and nuclear genes across different goldfish forms. The mitochondrial signal was clean and consistent. Every form of ornamental goldfish traced back down the maternal line to Chinese goldfish, Carassius auratus auratus. On the mother's side, the family tree was exactly what everyone expected, but the nuclear genes told a different story. In one major group, the common goldfish, the plain orange fish most people picture, three separate nuclear genes were not crucian carp genes at all. They were highly homologous to the genes of an entirely different fish, the common carp Cyprinus carpio. The goldfish on your windowsill, in other words, carries DNA from two different species in its nucleus. Somewhere in its breeding history, goldfish and common carp interbred, and the offspring of that crossing flowed back into the ornamental line. Read that again. The fish we treated as a single species, slowly refined from one wild ancestor, is, at the genetic level, a chimera, a goldfish mother's lineage carrying common carp genes spliced into the nucleus through deliberate or accidental hybridization. The breeders of imperial China were not simply selecting pretty mutants from a single pond species. At some point, they were crossing two different kinds of fish and folding the result back into the line. The body plan that looks so obviously like a crucian carp is concealing a second parent, and the two fish would have shared the same water constantly. Common carp were the great food fish of Chinese aquaculture, raised in the same managed ponds where ornamental fish were kept, so the opportunity for the lines to cross was always there. A few wrong eggs from a neighboring pond away. And here, the story becomes darker, because it reveals how little of the goldfish was left to nature at all. The 2020 genome study went looking for the specific stretches of DNA that human selection had hammered on hardest. These are regions called selective sweeps, where a favored trait dragged whole neighborhoods of genes along with it, as breeders fixed it into the population. When they isolated the most intensely selected 1% of these sweeps, they found just under 1,000 genes caught in the crossfire of human preference. And most of those genes did the same two things. They controlled pigmentation and body shape. 1,000 years of selection had concentrated itself almost entirely on how the fish looked, its color and its silhouette, with only a handful of changes touching behavior. The goldfish is, at the genomic level, an animal sculpted for appearance and almost nothing else. The color itself has a hidden mechanism worth pausing on. A wild crucian carp is grayish-green because its scales carry dense layers of dark pigment cells called melanophores. A goldfish turns orange, not because it gains a new color, but because it loses the dark one. The black melanin recedes, unmasking the red and yellow pigment cells that were beneath it all along. A young goldfish often hatches a drab wild olive and only later turns, the dark coat dissolving over weeks to reveal the orange underneath, a miniature replay of the change breeders first locked into the line 1,000 years ago. What looks like the addition of beauty is really the subtraction of camouflage. And a fish that has lost its camouflage is a fish that can only survive where nothing is hunting it. The DNA cannot tell us what each breeder was thinking when they chose one fish over another. It can only tell us where the pressure fell, and the pressure fell overwhelmingly on the surface of the animal. This is the deepest difference between the goldfish and almost every other domesticated creature. Dogs were shaped for work, then companionship. Cattle and sheep and pigs were shaped for food. The chicken was lured in by grain. But the goldfish was never bred to be useful in any of those ways. It was not bred for meat. It was not bred for labor. It was not even bred in the deepest sense for companionship. It was bred to be looked at. Because human taste in beauty is endlessly variable and arbitrary, the goldfish was pushed in dozens of directions at once, with no functional pressure pulling it back toward anything that worked. You can see that arbitrariness is written into the order in which the fancy varieties were created. Researchers reconstructing the sequence of artificial selection found that one of the very first things early breeders achieved was not a new color but a deletion. They fixed strains that were missing their dorsal fin entirely. The long ridge of fin along the fish's back, the structure that keeps it stable and upright in the water, was simply bred away because someone preferred the smooth, finless look. The genome study later confirmed how deep this ran, identifying roughly 400 genes associated with the goldfish's dorsal fin by sequencing egg goldfish, a variety that has no dorsal fin at all. 400 genes standing behind a structure that breeders erased on a whim. Only after the dorsal fin was gone did the breeders move on to the eyes. They began selecting for fish whose eyes bulged outward in grotesque telescopes, and for fish whose eyes rotated permanently upward, staring at the sky, the celestial varieties. The genetics revealed something almost unsettling about how this happened. A study from biologists at Tokai University examining a large portion of the mitochondrial genome across 44 goldfish strains found that the improvement of celestial eyes and telescope eyes took place independently at different times. The same monstrous feature was invented more than once, separately, because human preference kept circling back to it. The researchers concluded that goldfish had been subjected to strong artificial selection purely to meet the diversified needs of human preference in an unsystematic way. Unsystematic. There was no plan. There was only 1,000 years of people reaching into the water and keeping whatever caught their eye, however broken it would have been in a river. Strangeness compounded as the varieties multiplied. Breeders fixed a strain whose scales lost their reflective layer and turned to soft, pearl-like mat, then a strain with great fluid-filled sacs swinging beneath each eye, the bubble eye, a fish that can rupture its own face against a sharp edge of gravel. They bred the ranchu, the so-called king of goldfish, its back arched and finless and its head swallowed in a raspberry-like growth of tissue called the wen, a mass that can eventually swell over the eyes until the fish can hardly see. They bred the lionhead and the oranda to carry that same fleshy crown. Each of these was a deliberate choice to push some feature past the point of function, locked into the genome and passed down. Each one is now reproduced by the millions, a frozen record of a particular eye in a particular century, deciding that this swollen, half-blind, unstable thing was the most beautiful fish in the pond. There is one more layer that genetics exposed, and it concerns where exactly the ornamental fish came from. For a long time, it was assumed that the fancy goldfish of East Asia, and especially the elaborate Japanese strains, might trace to the local Japanese crucian carp. But when researchers built phylogenetic trees from the genetic sequences, adding 19 further data sets from DNA databases to their own samples, the Japanese goldfish did not group with the Japanese crucian carp at all. They originated instead from one of the two groups of Chinese crucian carp, the form known as the gibel carp. The entire ornamental tradition, wherever it later flourished, flowed out of a single Chinese source. The fancy goldfish of the world are not a scatter of independent inventions. They are branches of one Chinese lineage, carried outward and then bred into ever-stranger shapes far from home. Across the sea to Japan in the 16th century, onward to Europe by the 17th, where they arrived as rare luxuries handed between aristocrats, and finally everywhere, until the descendant of an imperial Chinese ornament could be won in a plastic bag at a county fair. So the deep-time goldfish, the hybrid of an ancient genome doubling, the maternal crucian carp line carrying common carp genes in its nucleus, is one thing. The animal in the bowl is another thing entirely, and it is much, much newer than the 1,000-year story suggests. The plain-colored carp of the Song Dynasty ponds were still recognizable fish. They had their fins, their normal eyes, their functional bodies. Everything that makes a fancy goldfish strange: the missing dorsal fin, the telescope eyes, the celestial upturned gaze, the egg-shaped body with no streamlining at all, the long trailing tail that slows the fish to a crawl. All of that was layered on top afterward, refined and multiplied over the most recent centuries as the ornamental trait exploded and breeders raced to produce novelties. And the genetic consequence of that race is an animal that exists at the edge of what a body can do. A fancy goldfish bred for a fat, round, egg-shaped torso has had its swim bladder crowded and deformed, so that many of these fish struggle to stay upright in the water at all, drifting and tumbling. A fish bred for enormous telescope eyes is half-blind, its bulging eyes vulnerable to injury and infection, navigating the world by smell because it can barely see. A fish bred to lose its dorsal fin has lost the very stabilizer that keeps it level. The ranchu, finless and archbacked, swims with a slow, laboring wobble that would leave it helpless in any current. When you look at a fancy goldfish, you are not looking at a pet that happens to be pretty. You are looking at an animal whose every striking feature is also a handicap, a creature that survives only because we have removed every danger from its world, in a glass bowl where there is nothing to escape from and nothing to catch it. And the cruelty of the contrast is that the wild original is nearly indestructible. A fancy goldfish escaped into a real river would die within days, while the plain feral goldfish, reverting toward its drab ancestor, has become an invasive pest on several continents, growing to the size of a football and choking out native fish. The toughness was never gone from the lineage. We simply bred it out of the ones we kept on the windowsill. What does this mean for the goldfish circling the bowl on your windowsill right now? It means the small, ordinary animal you may have won at a fair or bought for a few coins is one of the oldest works of living art on Earth. Its body is a hybrid built from two fish fused in deep time, its nucleus salted with the genes of a second species through forgotten crossings, its color and shape and ruined fins, the accumulated fingerprints of 1,000 years of human eyes choosing what they found beautiful. It was never bred to feed us or work for us or even keep us company in the way a dog does. It was bred only to be seen, and in being seen, it was reshaped into something that could not survive. The wild its ancestors came from. The plain crucian carp still swims, dull and tough, in the ponds of East Asia. The goldfish is what happens when 1,000 years of human preference is poured into a single living thing with no other purpose at all. In a glass bowl on a windowsill, a single orange fish turns in slow circles. We do not know which breeder first kept a red fish instead of throwing it back. We do not know which knight, in which dynasty, someone first crossed a goldfish with a carp and folded the strangeness into the line. We do not know whose hand first selected the fish with no fin on its back, or the one whose eyes had begun to turn toward the sky. But the descendants of all those small forgotten choices are still here, turning in the light, carrying 1,000 years of human longing inside a body that nature never built and could never have made on its own. If you found this story remarkable, consider subscribing. There are more origins to uncover, and the DNA is only beginning to tell us what it knows.